Hi guys, I thought that I'd do a quick recap on sources of genetic variation and try to explain it all to you. Firstly, before we get started, apologies in advance. I honestly can't bring myself to get off the couch this evening, and I have a dog next to me that snores something fierce and a husband playing Xbox. So any weird noises, hopefully that will explain all of them. Okie dokie, I'm going to try to simplify things and there's going to be a little bit of revision, but bear with me. The first thing that I need you to remember is that we have got 23 pairs of chromosomes, so that means we've got 46 according to my bad maths on a Friday night. Hang on, let me get you a picture. Okay, so let's pretend for the sake of the argument this is chromosome number 1, well at point 1, so you've got 2 chromosomes at point 1, there is 1 chromosome right about there, which is from your mum, and a second chromosome at that point there from your dad. Now what I want you to notice is that these chromosomes are both coding for the same genes. Okay, so what you've got here is these chromosomes they're both containing the same genes, so say this one here might be eye color, this one here might be hair color, just for the sake of the argument. So each of these 2 chromosomes at point 1 are both coding for the same genes, but what you'll notice here is that we've got some different alleles, so we'll get back to that in a moment. Alright, firstly though, remember that on any chromosome there are several different genes. Okay, like there could be dozens or hundreds. So this is clearly oversimplified, but you get the idea. So remember, if we've got, when we're talking about DNA and how it gets converted into protein through, mRNA being produced to going to the ribosome, etc., what was happening was that any particular gene was being read, so that might have been, say, this area here, and that gene is coding for a particular polypeptide or protein. Okay, so every chromosome codes for multiple genes, and at any point you've got 2 chromosomes coding for those same genes. And these 2 chromosomes at this one point are called a homologous pair or homologous chromosomes. So homo meaning same, these are chromosomes pairing for the same group of genes. Alright, so in our oversimplified diagram here, we have got 4 genes on these chromosomes. A, or the blue one, the pink one, which is B, the yellow one, which is C, and the red one, which is D. I thought I'd do it in colours and letters, just in case the colours don't come out properly. But you'll see here for the blue gene, this particular person is heterozygous. So they've got a big A. Oh, hang on. That wasn't meant to happen. You get a big A. Let's try that again. So you've got a big A here and a little a here. So this individual is heterozygous. They've got one of each allele. Again, here in the pink gene, you've got a little b and a big B, so they're heterozygous. And then down in the yellow and the red, you'll see that they are homozygous big C and homozygous big D. Okay, now I want to do just a little bit of revision about the process of meiosis. What you'll see here is prophase in this first couple of in that first line. You've got metaphase here, here. There we go. Sorry, technology getting the better of me. Anaphase here, and telophase here. Now remember, there is a time before prophase, and before prophase there was another time. Sounds like something for a movie. And what that was called was interphase. Now, at that late interphase, something really, really important happens, and that's when each of the chromosomes, each of the halves of the chromosomes actually duplicate. So this means that at the chromosome 1 point, you now don't have one copy of a chromosome from mum and one copy of a chromosome from dad. You've now got 2 of mum's copies and 2 of dad's copies. So going back to our previous example where we had our mum and dad chromosomes at point 1, we now have 2 at each like this. So we have 2 of mum's here and 2 of dad's over here. Now these are, as the picture would indicate, called the sister chromatids. Okay, so just to get your terminology right, over here, this is a homologous pair. These are sister chromatids. And this process of duplication has happened before the prophase stage in what we call interphase. Okay, so far, whilst this whole presentation was meant to be about introducing genetic variation, we haven't actually introduced any genetic variation yet. That was kind of a recap. So now we're going to talk about crossing over, which is our first part, or our first opportunity to have genetic variation. So crossing over happens after the duplication of these homologous chromosomes. So picture 1 now becomes picture 2, and what happens is that the tips of the homologous chromosomes, remembering the homologous chromosome was like this one here, and now these other ones can actually also interact, but that's kind of beside the point. What happens during this crossing over is that the tip of one of these can actually swap with the tip of the other half of the homologous chromosome. So what that ends up looking like is as follows. So this tip here swapped with that tip there. So whereas we had a big A with a little b over here and a little a with a big B over here, now when you come and have a look up at this diagram here, you'll see that on this part, this sister chromatid, we've now got a little a with a little b, which differs to the way everything started off. This one here is still the same because that one hasn't crossed over yet and it may never. And then we've got this one here, which is now a big A and a big B, whereas over here it was a little a and a big B. Now all of this occurs in prophase, which is broadly here. Remember, centrioles go to the poles, centromeres are created, and what happens is that all of these sister chromatids and the homologous pairs all jump on these spindle fibres. Now they're not lined up at this stage, they're kind of just haphazardly on there, but it's at this stage that we get this crossing over occurring. Okay, so you've got the homologous pairs that have duplicated right next to each other and they kind of twist up and the ends can snap off and glue to the opposite side. So crossing over happens during prophase. So our first source of genetic variation is occurring during this prophase stage. Now the way that this is creating variation is, if you can imagine, this is grossly oversimplified, but hopefully it will help. If you imagine that, I'll change coloured pens because this one's got plenty of scribbles all over it. If you imagine that, say, big A meant blue eyes and little b meant brown hair, we've got blue eyes mixed with brown hair. Wow, my handwriting's terrible. This is why I normally use the text feature. Whereas up here now, we've got our blue eyes mixing with, let's say the big B is for blonde hair. So it allows different combinations of traits. So you're mixing up the combinations of alleles. Okay, our next source of genetic variation that we're going to be discussing is the independent assortment / random segregation of genes. Okay, so before we were looking at chromosome 1 like this, after the duplication process had taken place. We're going to simplify this a little bit. So remember this was mum's halves and this was dad's halves. Gosh, this is getting even worse. I'm now trying to draw with my left hand. That was a bad idea. Okay, what we're going to do is simplify this and just look at this as though this was the mum's sister chromatids, these are the dad's sister chromatids, and just to be able to illustrate this a little bit better, we're going to be looking at a second chromosome at the same time. So that's mum, that's dad. So basically, the red and the pink, gender stereotyping here, are going to be female ones. The different shades of blue are going to be the dad-based chromosomes. Now, remember we did prophase and we talked about metaphase. We talked about interphase and we talked about prophase. Now we're going to get to the next process involved in meiosis. So before all of this meiosis started was our duplication. Then we had crossing over happening in our prophase stage. Then what happens is, you can see in this diagram here, you've had the crossing over take place. There's the little red and green swap tips. But this is critical now because they're all lining up along this equatorial plate. And this is where the next variation in genetic variation occurs. When we're looking at these two sets of chromosomes, so this is chromosome place number 1 and chromosome place number 2, remember there are 23 of these, so there's a lot of different combination possibilities. But what can happen when they are lining up during metaphase with this equatorial plate in place is that they can line up in a few different ways. They can line up as shown here with mum's chromosome 1 on the left hand side and mum's chromosome 2 on the left hand side of the equatorial plate. Or alternatively, they can line up with mum's chromosome 1 on the left hand side of the equatorial plate and chromosome 2 on the other side of the equatorial plate, so that if we come back and have a look over here for a moment, what happens is, what is about to happen anyway, is that you're about to have your cell split down the middle. So in this example back up here, dad's chromosome at point 1 is going to mix up with mum's chromosome at point 2. So when someone says, oh, you've got your mum's eyes, well, that's where mum's eyes came from. And here's your dad's chin. So this is how those different combinations can come to be. So the independent assortment is how they're lining up relative to the equatorial plate. And which one ends up on which side of it is completely independent of the way the next one lines up on that equatorial plate, which is completely independent of which way the third one ends up along the equatorial plate, etc., etc. So you've got 23 opportunities for various combinations. Now, I've been doing some research and I cannot find anything definitive. I believe that there was a past HSC question that asked you to distinguish between independent assortment and random segregation. Now, honestly, for all the reading that I've done, I can't find anything definitive on what that difference is. But if I had to be writing an answer to that, I would be saying that the independent assortment is which one lines up on which side of the plates. And then I would be saying that the random segregation is which way they split. Okay, so during that anaphase where they go to the different poles. I can't give you a more definitive answer on that. I am just going to have to say, let's hope that it never comes back. I can't imagine, it wasn't a fair question, so I can't imagine they'll ever get it passed again. But independent assortment / random segregation introduces variation because of the possible different combinations that mum's genes and dad's genes can line up at each of those 23 different points. So that will then determine which set of genes go into one cell on one side versus those on the other side. So your sources of genetic variation is your crossing over happening right about here, and at your independent assortment random segregation happening here. The last form, or last opportunity for, not the last, I'm sorry, the other source of genetic variation is mutations, which I won't go into here, but you've also got randomness introduced by which egg is ovulated, which sperm happens to swim there and make it first. So there are different aspects of randomization that continue beyond this point, but in terms of how it all works with meiosis 1 and things for you to be able to talk about, the three main ones in your course have been crossing over, independent assortment, random segregation, and your mutations. I really hope that this helps. I am got my fingers and toes crossed for you come exam time, and I hope that this has certainly made it clearer for you. Bye.